Hello, good people. I hope you are all safe and well in these tumultuous times. Whew. Just never know what's coming up next in this lifetime, do you? You know? Anyways, we'll have a lot of things to tell our grandkids about 2020 and 2021. Something to look forward to. So we're here to talk about detox. I'm here to say Happy New Year. And I just want to kind of introduce the format and what the heck I think I'm doing here, and see if that meets with what you think we're supposed to be doing here, and get this whole thing going. So I want to talk a little bit about like the overall format for this year. For those of you that may want to join in at different times, we've got our whole curriculum planned out for the year. And we've got a whole variety of ways you can interact with us. We've got these things like I'm doing today, this mini series, which is free. Free is good, right? Free doesn't mean that it's not good stuff. I put all my really good stuff in the free part, so free is fine. But and then we have boot camps that we're going to be doing. I'll talk about that later, which are priced around $1,000 plus or minus a little bit. Some of the boot camps are business oriented. Some of the boot camps are more clinically oriented. And then we have the mentorship, which is the big thing that I do for a select group of doctors that are really interested in this kind of work. We take them on and train them, quite thoroughly, I think, in how to interpret labs. So most people start off in this arena that we're in functional medicine, being really curious about the labs, but it's hard to find solid and really great training. And I've been very blessed since the very beginning of my career in 1992, 93, to just have this series of mentors that trained me. And that continues to this day. I'm, I don't know, almost 30 years later, I work now twice a week with the greatest scientist in our field, Dr. Richard Lord. And he and I have been collaborating for 4 years now, twice a week, many, many hours every day, to really let me understand the origins of the lab work. So Richard's the scientist that developed what we now call organic acids and fatty acids, which turned into things called like the organics profile and the NutriVal profile. And as the scientist that developed this work originally, many decades ago, his insight into these tests is pretty profound. And so he and I have developed this collaborative relationship. It's kind of like a father-son thing. Richard's in his late 70s, almost 80 now, and he's just trying to teach me everything he ever learned so that I can teach my audience of students. And that's what we're going to talk about today. So all the things I'm learning from Richard right now. And obviously this work has then also infused itself into the mentorship courses. And so when I started the Kalish Method mentorship, it was like about the Kalish Method, and who knows what that even was. That was pretty simple, dimple stuff. It was, adrenal or is adrenal and GI material, and it's good material. I mean, it's something that you should learn if you don't know it. But now what's happened in the last 4 years is I've been able to really take my teaching skills to a different level because I have Richard chiming in my ear, twice a week, just dumping thousands of little factoids into my brain. So now the Kalish Institute courses are really quite different than they were even a couple of years ago in terms of me really just being a spokesperson for Dr. Lord's work. So we're going to talk today briefly about detoxification in clinical practice. And I really want to cover the things that I didn't really know, that I thought, I knew a lot about detox, but I didn't. And so I want to cover those things. Not the stuff that you already probably know, okay? I'm going to cover, and these are not unusual things. These are common things. They're common for patients, but they're not just commonly taught. So right off the bat, we're going to talk about ammonia clearance. Who even knows what that means? I mean, that's a common problem. You see patients that are toxic in ammonia. We'll talk about everything from ammonia to xylene, and a lot about low glutathione and metals, and how you can really start to jump in and understand how these detox pathways fail and what you can do about them, and how much of this is just really related to genetics. We'll talk a little bit about magnesium and detoxification, and we're going to get into two subjects in the most detail that I want to focus on, and the one is cell membranes and how cell membranes work and why cell membranes really matter and how they, to a certain extent, can very strongly determine toxicity levels in patients and how cell membranes can get messed up. And then we're also going to talk a lot about proteins and protein folding and how the protein folding process, if it's not done well, can cause some major problems. So just lodge that in the back of your mind somewhere. And we'll talk short talk today, just like 20 minutes max. And then I want to look at some cases so you guys can go out and enjoy the rest of your day, okay? And so for those of you that are new, this is me. I'm Dan Kalish. I've been in practice for a long time, and we've been working with IFM for a while. I've done some research projects, but for most of what we're talking about tonight really is about the work that I've done with Richard Lord. We have a couple of classes that are coming up soon, starting in January. Well, it is January. Starting at the end of this month, January 25th, we have the mentorship where we're getting into all the advanced testing and the functional genomics work that I've learned from Richard. And that's a 1-year-long program, small group format. We have small groups of doctors, and each week we review labs together. There's a very extensive curriculum. We keep these groups pretty small. It's a pretty elite group of doctors. You get $1,000 off if you're interested and you're listening to this. There's the code there, VIP Jan 21. And if you're curious, you can schedule a call with my salesperson or with myself, and we can talk to you more about that. We also have a series of boot camps that are going on this year, and there's going to be 4 of them. Count them, 4. There's this one coming up on business. Then there's going to be a clinical one a few months later on lab interpretation, and the boot camp coming up February 8th is on telemedicine, telehealth, 8-week deep dive into how to do some of the business side of this. All right, so that is the announcements. And in general, when I think about this, by the way, as a caveat, for those of you that are counting slides, there's like 50 slides in here, and we're going to cover like half of them. But I just wanted to put some of these in for dramatic effect. So I know I'm going to slip through them quickly, but hopefully it won't bother you too much, okay? But 2 types of patients, easy to solve. You just tell them to stop drinking alcohol, do like a detox supplement over the counter, and then they just miraculously get better, right? And so for those people, you can do non-lab-based programs. We're not even worried about them because you can just do some generic detoxing with them, and they're just going to feel better, and then it's all fine, right? Well, we're talking about people that have really major problems, and that want to always, contextualize this, and that we're talking about detox tonight, but we always work with the neuroendocrine and GI systems also in an integrated way. It's hard to teach all 3 at the same time because you wouldn't be able to, in an hour, but just to acknowledge that, okay? And now I'm going to skip through these slides really fast, but it's just to point out the features that there's a huge amount of environmental toxins, and every single one of our patients has 100 to 200 to 700 toxins. There's 80,000 things that we've developed that are chemicals that are really bad for us. And, every baby that's born in the United States now, according to the CDC, has somewhere between 50 and 70 toxins the day they're born. So this is not a problem just of some people. This is a problem for everyone that we're working with. If you really want to learn more about it, look at the CDC's 4th report. You can Google that. It's a couple thousand pages of the government reporting on human exposure to environmental chemicals. And I have a 1,500 PowerPoint slideshow on this, and we're just going to skip through these just to say that there's a lot of bad stuff out there. And I don't want to spend time, and a lot of them for some reason have these initials that are like 3, like BPA, DPD, DDT, all that stuff, right? This is all the bad stuff. And you can be sure that almost everyone you're working with, unless they've done their own major detox, is soaked with chemicals and heavy metals from the home, from the food, from the wherever it may be, okay? And anyways, that's just kind of to prove the point. And in the order of importance, we're super worried about heavy metals, pretty worried about biotoxins, kind of worried about xenobiotics, and somewhat worried about food preservatives. So even if you're eating organic all the time, the main problem for most people is going to be around the metals and chemicals, and not necessarily just from food, right? So it's pretty much impossible to avoid any of these things now. You just can't. And then when you think about symptoms and who are the patients that we're talking about, it's everybody, okay? This is why everybody's tired and everybody's overweight and everybody needs ADD meds and everybody has trouble focusing and everybody drinks tons of coffee because everybody that we're working with is toxic. And if it gets really bad, people get super depressed, bad things happen. And for most of today's talk, really, we're going to be centered around the liver because there's 3 ways you can detox, right? Through your sweat, through your urine, obviously the kidneys and the urine, or through the liver dumping stuff out into the intestinal tract. And we're going to talk mostly about your liver and a whole bunch of different pathways. The ones that we're all the most familiar with, right, are phase 1, phase 2. And in fact, when you think about detox, that's usually what most people think about. But what we're concerned about in general, or concern is not the right word, kind of focused on in general, is how we're going to be able to measure for toxin-related problems, right? And I want to show you guys at the end of the session today, we'll review some labs, how you can tie the detox pathways that we've learned about so frequently back to the labs. So you can measure with the labs that we're going to look at tonight. B vitamins, very specifically. You can measure glutathione and antioxidants and vitamin C and vitamin E. You can measure glycine and taurine and glutamine and cysteine and N-acetylcysteine and methionine. You can measure every one of these things with a lab. This lays out every aspect of cytochrome P450 enzyme function. That's what we call phase 1. You can lay out every aspect of phase 2. The fact that you can measure this stuff is just phenomenal. I mean, I just sometimes have to pause for a moment and just think what a bloody miracle it is that we can do that, you know? And if you're even remotely interested in integrative medicine or natural health, you should just learn how to do this, because it's just for your own entertainment, if not for the sake of humanity. It's just fascinating kind of work, right? And the fact that everyone that you're talking to every day in your practice has got between 100 and 700 toxins, you just got to learn how to deal with this as a, you know, in an intricate and complex and profound kind of way. You got to really dig in and learn this work. So we'll talk a little bit about phase 1, cytochrome P450 enzymes. That's coming up. And we'll talk a little bit about phase 2. So the nutrients that are involved in phase 1, you can see them listed out here: the B vitamins, the antioxidants. Phase 2, you get into the sulfur amino acids. So basically, your body takes a toxin, it makes it, it grabs onto it with phase 1, starts to break it down to an intermediary. Then phase 2 binds it up and gets it water soluble, and then it comes out through the stool, the urine, or the sweat. Okay, but if you're not doing well with any one of these nutrients, or you're not doing well because of diet or genetics, then you're going to have a pretty serious problem with impaired detox. So it can be just simply because you're overwhelmed with too many toxins, and these fat soluble toxins just build up in the fatty parts of the body and they get stored, and then all of a sudden the patient's tired, depressed, or anxious, they have skin problems, all this is going on. Right? So we want to clean this whole system up, both on the inside and outside. So cell membranes, we're going to talk about for a few minutes. Then we're going to talk a little bit about amino acids. So kind of open your mind to some of these primary nutrients that are critical for detoxification. So why do cell membranes matter? It's a membrane, right? It's keeping things out. That's what it's doing. It's like if you have a castle and the castle has like a moat and then a bunch of walls around it. That's exactly what your cells are like. They have a dual layer, biphospholipid layer, right? They have this double layer of fats that are keeping bad stuff out. Similarly, you need to get toxins that are made inside the cell out through the membrane. So the fluidity and structure of the membrane is the key to its function. And it's somewhere between olive oil and Jello. So what I mean by that, these are made up of fatty acids, they're fats. So if you like go home tonight and you make some salad dressing, you pour some olive oil and you spill it on the counter and you wipe it with your hand, it's like oily, right? Those are fats. So our fatty acids in our body at body temperature are fats, right? They're not like margarine or something that's a solid fat, right? They're oily. And there's new research that I was just reading the other day that says, okay, maybe they're not as oily as olive oil, but they're kind of maybe a little more the consistency of Jello. But point is that the membrane is moving and fluctuating, and it's not a hard, rigid structure by any means. If you think about how hard and rigid a fat is, like olive oil, that's kind of what we're talking about. Cholesterol, importantly, stabilizes these oils or these fats, right, to make the membrane more robust, to make it sturdier, to make it stronger, basically. And then there's proteins that are stuck in the membrane that are responsible for its functions. The membrane itself is just like membrane, right? It's not doing a lot. It's the proteins in it that are allowing things to go in and out of the cell in really specific ways. Of course, some things are diffusing through the membrane, but we're mostly worried about the things that are being transported through the membrane, okay? And it's mostly made up of phospholipids. Okay, and it's a nice little quote here. Cell membrane, or the plasma membrane, surrounds every living cell, delimiting the cell from the environment around it. Okay, that's what it does. And here's what it looks like. And remember this from school. There's this one layer of fatty acids here. There's another layer of fatty acids here. And here's a protein that's embedded in those fatty acids. So if the membrane's fluid and flexible and movable and the fats in there are super healthy, good fats, then it works. And if the membrane is made up of fats that are not healthy, that are brittle and old and crappy quality fats, then you have a brittle and old and poor quality membrane. And then what happens? Stuff gets in that shouldn't, and you can't get stuff out. Okay. And there's another way of looking at the membrane. It's a phospholipid bilayer. It's these two fats that are lined up kind of against each other, right? Because they're hydrophobic, right? The fats are trying to get away from the watery part of the cell, so they're lining up like this. And there's a little phosphate group on the end, and the fats are kind of dangling in there between each other, trying to stay away from the watery parts of the cell and from the external environment. Okay, and then within this membrane, we inject little chunks of cholesterol. It's the brown stuff there, which gives some structural integrity. And that's one of the major and most important roles of cholesterol, is to help maintain the strength and integrity of these membranes. Now, let me blow this up. When you're doing lab testing, one of the ways, there's many ways to look at the health of the membrane, but one of the ways you do that is by measuring the omega-6 and omega-3 fatty acids. And if the omega-6 or omega-3 fatty acid levels are not good, it means you're going to have poor quality cell membranes, because you're making the membranes up of these exact fats. Okay, so from a testing perspective, we can directly measure these fats, which tells us a lot about the health and functioning of the cell membrane. Okay. And now, so that's part one, right? Essential fatty acids make up the membrane. Membrane keeps toxins out and allows toxins that are made inside the cell to get out, so it's controlling the flow of toxins. Great cell membrane quality means you have less toxin-related problems. Bad cell membrane quality, you're just a target for every little neurotoxin that's walking around out there, okay? So now, we just saw a picture, and this is right here, of these proteins that are embedded in the cell membrane. Okay, protein is made up of amino acids. Okay, and one of the proteins that's in the cell, as in a membrane, right, is cytochrome P450 enzymes, which are phase 1 detox, right, which just looked at a minute ago. So it's a membrane bound protein. It's hard to say, membrane bound protein. And here's a picture of it. It's a beautiful structure. This is the actual structure of cytochrome P450. It's beautiful, isn't it? Intricate. Each one of these is just trying to depict all these amino acids that are floating around. And so now, the more complicated that a protein is, and as you can see, this is a pretty darn complicated protein. There's a lot going on there. The more susceptible it is to genetic defects because of something called protein folding. So when we are making our proteins, like enzymes that help us detox, what we do is we grab all these amino acids, you remember this from school, and you string them together, and then eventually they can turn into like this alpha helix thing or into a beta sheet, and then that string of amino acids folds itself in really beautiful ways and ends up ultimately creating a protein. Okay, a string of amino acids binding together based on the electrical charges of those amino acids. They fold into a certain configuration after going through these steps of turning into beta sheets and alpha helixes and all these different things. And you guys all learned this in school a million years ago. You probably forgot most of this, but it's pretty important, because if that series of amino acids is a little bit off, one of these is out, and there's one that's in place that's in the wrong place, that's going to change how it folds, how the protein folds. And if you have a SNP, what that means is that there's one of these amino acids out of sequence, right? And if one of those amino acids is out of sequence, when the protein goes to fold, the charges aren't going to be lined up in the right place, and it's not going to fold very well. And that means that the enzyme, in this case, a detox enzyme like cytochrome P450, is not going to work very well, which means that if you're exposed to even a small amount of toxins, you're not going to be able to clear it. Okay, and here's some other examples of proteins. So let me just say that one more time. We take amino acids, we string them together. If it's all working right, it folds into a protein, and then the protein does stuff. Like in this case, get rid of toxins, right? If the amino acids are even slightly out of sequence, then the ability of that protein to fold, it's not going to fold right, and it's not going to function right. So the more complicated the protein is, the more likely there could be a problem. You just saw a second ago how complicated cytochrome P450 is. It's an enzyme that's a protein, right? That's membrane bound. So it's dependent on the health of the membrane, and it's dependent on the amino acid strings, right, to all work properly. So guess what? If a person is low in one essential amino acid, you can't you can't make these proteins, right? Let's just say that one more time. If you want to make a complex protein like this, you have to have all the essential amino acids present. And if you're missing an amino acid, you can't assemble a protein. So if amino acid levels are off, you're not going to be able to make these structures. If the genetics are off, you're going to make these structures wrong. So it turns out amino acids are really important, not only for their very specific functioning in terms of running phase two. So amino acids run phase two. That's a very targeted, specific function. But amino acids are also important because you have to have amino acids to make proteins. All these enzymes we're talking about for detox are proteins that are entirely based upon amino acids. So if you're missing an amino acid, you can't make the enzyme. Okay, and then separate from that, we have these amino acids that are specifically for detox. So you need amino acids to make the ability of detox to work, and then once detox pathways are working, you need the amino acids to keep them going. Okay, so that's my just short introduction. Fatty acids are super important, amino acids are super important, and if fatty acids don't get converted properly, that can occur because of genetic issues, and if amino acids are not stringing together properly to make enzymes properly due to genetic issues, you're going to have enzyme problems that aren't working. All right, so I'm just going to take a deep breath and relax for a second after that. And now, that's the end of part one. Now, I don't know, you can check your phone or something, or check your email for a second. We're just going to take a short break here for me to think about life. Okay, now we're into part two. You ready? All right, now we're going to look at specific stuff. So by specific stuff, I mean like a specific pathway. And here's one on the board, the urea cycle. Why is this important? Well, the urea cycle gets rid of ammonia. Ammonia can build up, and ammonia is not the kind of thing you want to have build up in your body and your brain. Take it from me. It's a really, I mean, not that I've ever experienced it personally because I haven't. I don't go around drinking ammonia or something, but I've had a lot of patients with ammonia toxicity. And so you can see here, there's a bunch of enzymes and they're conveniently numbered. And you can try to pronounce these if you want, but anyways, there's 6 of them, okay? Let's just call them 1, 2, 3, 4, 5, 6. These are enzymes. What do enzymes look like? Well, we just saw one. Enzymes look kind of like this, don't they? Okay, they're proteins and they're strings of amino acids. And we just said how enzymes, the more complicated they are very prone to being made improperly because of genetic defects. So just imagine you have a cycle like the urea cycle that has 6 enzymes. There's 6 different enzymes that could get messed up here from a SNP. That's a lot. And there's multiple ways that each individual enzyme could get messed up. So you are often going to see a defect in the urea cycle of your patients that's causing an ammonia toxicity problem that's going right into their brain and causing brain fog and fatigue, and neurological problems, and all that kind of stuff. And yes, you can measure this on a lab test. And we'll look at these in the third part of today's talk. You can measure orotate. If that's high, there's a problem. You can also measure the individual aminos like arginine, ornithine, citrulline, etc. There are a bunch of ways that you can measure this and see if there's a problem. We'll show some examples in a minute, okay? So anyways, common thing that's wrong with your patients, if you don't know about that, it's kind of important to figure that out. And let me see here. Okay, so we're going to look at, we're going to cut over to labs in a minute. We're going to look at examples of phase 1 and phase 2 problems. So just in the back of your mind, remember about B vitamins, antioxidants, and sulfur amino acids that makes up phase 1 and phase 2. And we're going to look at some examples of urea cycle defects on labs as well. And then we're going to look at examples of fatty acid related problems. I kind of glossed over this one kind of fast, but remember, because why do fatty acids? Remember, are important because of cell membranes. We're going to look at omega-3 and omega-6 fats. And you can see here, these fats are go from one fat to the next based on these enzymes, right? You've heard of desaturase and elongase enzymes, okay? They're the ones that are converting one fat to another. Enzymes, you might guess, are proteins, you might guess, are made from strings of amino acids, so they're prone to genetic defects. So a lot of your patients are going to have problems with the enzymes that help them produce the various essential fatty acids that are critical for the cell membrane. Okay, so cell membranes, fatty acids, amino acids, those are the things I want you to keep in mind. And then we'll talk a little bit about solutions here before we look at the labs. None of this is that complicated. You know, you want people to drink a lot of water, lose a lot of weight, exercise a lot. You know, all the basic lifestyle stuff is essential for detoxification. There's nothing particularly complex about that. It's just a matter of getting people to do it, you know. And then just as a quick reminder, you get $1,000 off the mentorship. If you see this talk, VIP Jan 21, we're starting a new group at the end of January. We're going to do a boot camp on February 8th. It's a business boot camp centering around telehealth and business essentials. That's an 8-week deep dive into learning how to tweak your business and kind of flip it towards more of a telemedicine, telehealth module. The mentorship is a full year of lab interpretation and hundreds of hours of training that will get you up to speed on all this good stuff. Okay, so now we're going to pause for another moment and let's look at some labs. All right. And you know, I honestly don't spend a ton of time scanning these labs ahead of time. And in fact, at about 2 minutes before class started, I just went into this lab folder and I just grabbed like 4 or 5 examples. So I don't know what we're looking at here, okay? Hopefully they're going to prove to be helpful. If not, it's going to be a little embarrassing, but and these are all patients of mine, so they're all kind of messed up. It's unlikely that we're going to find a healthy one here. Okay, so here are detoxification indicators. This is an organic acids profile from Genova Labs. So right off the bat, you're looking at these like that looks normal, that looks normal, normal. I don't know what that one means. You can't detect it, but that one looks normal. And then like, uh-oh, wait a minute, sulfate is low. So my teacher, Dr. Richard Lord, who's the scientist that developed this test, told me on one of our very first sessions 4 years ago, the most important marker on this entire lab, not on the detox part, the entire lab, the most important marker on this entire lab is the marker for sulfate. Why? Because when sulfate is low, it means the patient is low in glutathione. And low levels of glutathione are incompatible with human health. It's a very, very important marker. Okay, low sulfate means low glutathione. That's low sulfur containing amino acids. That's poor detoxification capacity. And it's crippling for a whole variety of reasons. So just keep that in mind. And low sulfate is a little clinical pearl thing, is often paired with low homovanillate. Homovanillate, many of you know, is the marker for dopamine. So if homovanillate is low, it implies that dopamine levels are low. Dopamine is made using sulfur amino acids. You have to have sulfur amino acids to make dopamine. Sulfur amino acids are used to make glutathione. I personally think it's a design flaw. If someone had asked me, I would not have set it up this way. No one asked me. I just stumbled upon this. hundreds of thousands of years after humans developed. And so we're just stuck with this. Sulfur amino acids are used to make dopamine, one of the major brain chemicals. The same exact, not different, the same exact sulfur amino acids are used to make glutathione, which protects the brain. So if sulfur amino acid levels drop and glutathione levels drop, your brain protection has dropped and your dopamine levels have dropped at the same time. It's a design flaw. It's not a great idea, right? Whoever set this up. It's a vulnerable position. You will often see patients like this one with low sulfate and low homovanillate. That means that their dopamine is washed out and they're toxic. It means there's a lot of toxins circulating in the brain potentially, and there's low dopamine levels. That's a very bad combination. People don't feel very good when they're having that going on. Now, I'm just scanning through these because that's what we do. Let's just see if this guy had any other problems that are obvious. Well, okay, here we go. It's not bad. So omega, we're also looking now, remember cell membranes, what are they made up of? Omega-3 fats are super important for cell membranes. Here we have our polyunsaturated omega-3s. Eicosapentaenoic, also known as EPA, EPA is low. If you have low omega-3s, you're going to have poor quality cell membranes. So that's going to lead to even a small number or small amount of a toxin getting into the brain and causing problems. So right off the bat, what would you want to do? Give the person omega-3s, give them EPA. Not a little bit either, give them a lot, because you want to get the cell membranes to repair quickly. Don't underdose people. Like a gram of fish oil is not going to fix like that kind of thing. All right, let's see here. Let's go to another one. We're doing pretty well on time. Oh, here's another case, 67-year-old male, omega-3s, alpha-linolenic, eicosapentaenoic. You can see all three of these here are low. So this person has an even bigger problem with their cell membranes than the last person. So toxins are just shooting right in past the membrane into the cell. And when a toxin gets inside the cell, there's all kinds of bad things that can happen. It could be damage to DNA. It could be damage to the mitochondria. It could be damage to the lipids themselves, right? And so if you have damage to DNA, puts you at high risk for some pretty bad diseases. If you have damage to mitochondria, that's not a very good thing to happen either. And if you have damage to the lipids themselves, you've got a problem. And things like heart disease, cancer, diabetes, this is all directly related to the ability of cell membranes to protect us against toxins. Okay? So anyways, that's that. Oh, and then we're supposed to look at amino acids. Remember, because that's on the lecture thing there. Okay, now this person has high taurine. Oh, okay, this person has several amino acids that are high. So when you see an amino acid level that's high, does that mean they eat too much protein or well, I mean, it could be that they're taking too much of a given supplement, but if there's individual amino acids that are high, what you're concerned about is that this is a blood test, right? And that those amino acids are circulating in the bloodstream, they're not getting into the cell to do stuff. Okay, and taurine's a sulfur amino acid that's required for detox. So you can have enough taurine in the system, it can be circulating in your blood, but not getting into cells when you need it. And how do you get amino acids to get into cells? That's a good question. I'm going to just ask that as a question, see if anyone can type in the right answer. How do you resolve a high amino acid level and get that amino acid to get into cells? Anybody know? Well, no one's answering. B vitamins. B vitamins catabolize, break down amino acids. So if you see high amino acid levels, you start to think about giving the person B vitamins so they can break them down. That's a good tip. Now, let's just look here. Okay, so now these are the detox markers on the next patient here. Sulfate on the last one was low, if you remember that. Sulfate on this one's a little high. Oh, Yeah, it's a little high. So that shows an increased utilization of glutathione. The person is burning through glutathione a little too rapidly. So you want to kind of hunt down and figure out why that is. Low sulfate, you use, for low sulfate, you use N-acetylcysteine, or you can use glutathione itself. Okay, one more case here, and then I'll get to questions at the end, okay? So let me just refresh for 1 second so we're all on the same page. Here, Urea Cycle, remember, is how we clear ammonia, and that's reliant upon arginine and some other amino acids, very susceptible to genetic defects. Not enough arginine, ammonia builds up, and this marker called orotate will go high. And then we talked a little bit about the cytochrome P450 enzymes, but the important part is that it's an enzyme, it's a protein, it's made from amino acids, very prone to having genetic defects. And you can have 100 people all eating the same amount of tuna fish every day, and the one that has defects with their cytochrome P450 enzymes is not breaking down the metals and is going to have a big problem. And then we talked about the importance of cell membranes, right? And that the membranes are important because of the actual fats and the quality of fats here. And the membranes are important because we have these proteins embedded in the membranes, okay? And the proteins are made from amino acids. So now we're going to look at another example. Remember, this whole structure here that's doing all this work is a protein, and it's made from individual amino acids. It's the only way you can make it. There's not some alternative path here. Now, look at this. Check this out. Wait a minute, I think I'm on the wrong one. Yeah, here, how about this? So you see, these are the essential amino acids from an ion panel, and you can see that lysine is borderline low, leucine is low, borderline low, valine is low, borderline low, phenylalanine is outright low. There's a whole cluster of these that are in the low ranges. Okay, so when you have a low amino acid, that's an essential amino acid. See the words there? Essential amino acids. It's not like an optional amino acid. It's not a when you feel like you want to have it amino acid. It's essential. So when they say essential, what does that mean? It means that it's essential to building proteins. So if we take one amino acid on here that's low, I don't know, pick one. Anybody have a favorite here? I don't know, phenylalanine, just randomly, okay? So when phenylalanine levels are low, what does that mean to the ability of the body to make proteins? Can you make some proteins without phenylalanine? No. There's not a single protein in the human body except for collagen. Collagen's weird, but let's forget about collagen, okay? Except for collagen, there's not a single protein in the human body that you can make unless you have phenylalanine. Not one. I mean, it's not just humans, it's like birds and amoebas. You can't make any protein that's needed for life except for collagen without phenylalanine. That's why it's essential. So if phenylalanine levels are low, forget about what it does for your brain. You can't make proteins. And what are these enzymes that we're looking at? The enzymes for the urea cycle, the enzymes for cytochrome P450, they're made out of protein. So if you don't have phenylalanine, you cannot make the enzymes that you need to run detoxification. That's why amino acids are really important. Okay, so there's two separate subjects here. Amino acids that are essential to make all proteins in the body, and then specific amino acids that we use for detox. I'm trying to separate these two out. Okay, unfortunately, the lab does this for us. So like we're on essentials now, but if you look at the next page here, God bless Richard Lord. I can't tell you how happy I am when we talk every week. And I get to talk to him twice a week, guys. It's like the most phenomenal experience. I don't know what I did in some past life to deserve this, but so anyways, they group together the sulfur amino acids, the glutathione related ones, right? So those are specific. So amino acids can do specific things and they can do broad generic things. Now, in this particular patient, we're looking at the urea cycle. Okay, this is for how the markers for how you clear ammonia, and you can see 3 of them are either low or borderline low. So that's a urea cycle that's not working very well. And it's a pretty simple solution. You give arginine. Some people also use citrulline or ornithine, but I usually use arginine depending on the patient, and that'll bring the citric acid, bring the urea cycle back and start to have that person able to clear ammonia. Just don't underdose them. 1,000 milligrams of arginine is not going to do anything, okay? You got to give a fair amount to make this stuff work. Now you're kind of getting used to this, right? It's like the third time. Low essential fatty acids. What does that mean? Cell membranes not so great. Toxins getting in, not getting out. Big problem with detoxification. Solution, give them some omega-3s. This person happens to also need some omega-6s. Just a little bit, right? See that? Okay, now let's look down here. Now this marker on an organic acids profile, 2-methylhyperate, is high. That shows that there's a buildup of xylene. And just as a reminder, here's our orotate. In this case, it's not high. If orotate is high, then there's a problem with ammonia detoxification. It doesn't always go high, but it can. Okay, one more case, and then I'll look at questions, okay? Oh, here's a good one. Okay, so now, without going any further, what do you see? Marker number 1 is low, borderline low. That's lysine. Lysine is an essential amino acid. Without lysine, you cannot make proteins. So right after, I mean, how complicated is this? You just give the person lysine, they're going to be able to make proteins, okay? That's not too complicated. Oh, and look at this one. A whole bunch of high amino acids. Means they're circulating in the bloodstream, means they're not getting in. Whereas some of them are low, like arginine, okay? Arginine's low, these others are high. You give vitamin B6 to allow the body to metabolize or catabolize or break down these amino acids and pull them into cells. And then let's look over here at the detox markers where they group it together here. So on the urea cycle, wow, that's a little wacky, isn't it? So low and high, and then kind of borderline low and then high. I mean, how could that even happen? Remember, the arginine is converting to citrulline, is converting to ornithine, is converting to glutamine. So when you see these dramatic breaks like this, where there's plenty of this substrate, but then this, there's nothing there, then there's clearly some kind of problem, right? And this person would benefit from taking arginine to get their detox pathways working better, and from B vitamins to get these amino acids processing better. So this person is going to be super high in ammonia, right? Because it's not working. The ammonia clearance system isn't working very well. Okay, this is number 4, and these are, I didn't even pick these on purpose, okay? Low omega-3s. You can't give omega-3s by themselves without doing this test though, because if you give omega-3s for a long time, you're going to lower omega-6s. And I see this in my practice every week or two. If you give omega-3s for a long time, they're going to compete with these enzymes that I showed earlier, and the body's going to become deficient in omega-6. So there's a lot of patients out there who take omega-3 and they make themselves deficient in omega-6. And that's going to screw up your everything, your prostaglandins, your inflammatory response, all that stuff. So don't let people just go out and take a bunch of supplements without doing the testing. That's insane. It's dangerous. It's better just to eat healthy food if you're not going to do the labs. Okay, and this person has good detox markers, so that's kind of a nice positive note to end on. Oh, but they have a yeast overgrowth. That's a gut problem. Okay, so I'm going to wrap up briefly here. Let me just review the concepts and see if this is starting to sink in a little bit. So number 1, we talked about phase 1 and phase 2 detoxification, B vitamins, antioxidants, sulfur amino acids. And we're pointing out very carefully that the amino acids here are very important for detoxification, but that essential amino acids are even more important because they regulate enzymes because they are what we make proteins from. Okay, so to detox, you need B6, B2, B3. We measure all these things on the labs. We need antioxidants and we need sulfur amino acids. You can measure all those things. And then we also are concerned about cell membranes and how that plays a role in toxicity, hence the omega-3, omega-6 obsession. And then we're a little bit concerned about amino acids because that's how we make these proteins. So I just missed one thing here and I want to show you real quick and then we'll look at questions. It just didn't show up on any of these examples, but I'll just show you on the lab. So I said earlier, just a second ago, that we're very concerned about B vitamins with detoxification. So on these tests here, you see B complex markers. They're measured all here in great detail. We just didn't really talk about it with these cases because they're not positive. So the B vitamins are measured very carefully. And then you can also measure oxidative stress and the need for antioxidants with this part of the test here. So these are the phase 1 kind of things, right? B vitamins, antioxidants, and then the sulfur amino acids are measured here. Phase 1 and phase 2, all these markers here. This marker here is for ammonia. There's phase 1 and phase 2, and these 3 here are all for glutathione. So once you learn how to interpret these labs, you can get to an impeccable level of specificity with your treatments. And it's fun. Not fun's the right word, but yeah, it's kind of fun. It's fun to fix people. So reminder, we got a mentorship class starting at the end of the month. You want to do this nonstop, day in and day out for a year and really learn lab skills to be at the super highest level of anyone in our profession. Come join the mentorship. Get $1,000 off if you enter that code. Small groups, tons of lab review, a million gazillion lectures. I talk about Richard Lord's work every week. It's pretty cool. Great group of doctors as well. I mean, even if you don't like me very much, or you think I'm kind of like a jerk, or I'm not very funny, the other doctors in the class do most, half of the talking, right? Because they're presenting cases and they're chiming in and they're saying, you know, this and that. So you learn a lot from the other practitioners too. And then we have a boot camp if you want to work on your business, February 8th, telehealth boot camp starting. Or we do a deep dive on the business stuff. Okay, now we got a few minutes left, and let's look at questions. So let me just go up to the beginning. If orotate is high, will you still give arginine if arginine is normal? Absolutely yes. So if orotate is high, 1,000 milligrams of arginine is not going to cut it. You want to be somewhere between 3,000 and 6,000 milligrams a day of arginine. Start low and taper up, but 3 to 6 grams a day. So there are sort of generic detox programs you can do too. I'm not that interested in those, but you can do those. Yeah, if someone's fatty acid levels are off, then they need to stop taking whatever the wrong fatty acid is. You're going to see this all the time. People take too many omega-3s all the time. You got to test fatty acids and get that ratio right, okay? You absolutely have to do that. That was Susan's question. If someone has ammonia toxicity, this is Elizabeth, can you smell it on their breath? Occasionally. And what's even weirder is occasionally when you give them a boatload of arginine, it starts to come out. The ammonia comes out in their sweat, in their urine, and they'll come back and they'll tell you, I'm smelling like ammonia. But that doesn't happen that often. But you know, I had a patient, that was about 2 years ago now, and I really like this woman. She's super cool. She was like a combat pilot, U.S. military trained combat pilot, which to me said that she was not like wimpy. She was badass. She was like serious person, right? And she was living at her parents' home because she was just so fatigued. I guess we would call it chronic fatigue, whatever word you want to use for it. She couldn't function anymore. You know, she was a military pilot at first and combat pilot, and then she became a commercial airline pilot, and then she had to just stop working, living at her parents' house, like in her early 40s, because she was so exhausted. Her main problem, really, I mean, pretty much her only problem was massive ammonia toxicity. Put her on 3 grams of arginine, Nothing really happened. I put her on 6, things started to move, and every week that ammonia came out, her brain got clearer and clearer. She got to be up and around. And the last time I talked to her, she was getting ready to go back to work. I mean, this stuff is pretty serious, and some people get screwed up on this. That ammonia problem is something you really want to be able to catch, okay? Let's see, B vitamins. Oh, if you do the ion panel where you get the blood drawn from, yeah, you can get any phlebotomist to do it. You can contact Genova, and they'll give you some ideas. But my office, we have never had a problem getting the blood drawn. It's pretty simple. And the patient gets their blood drawn from any phlebotomist anywhere, just FedEx the kit back to the lab. So if you have low 3s, I usually do the low omega-3s. I usually do a loading dose. So I'll give them 4 grams a day for a couple of months and then cut it back to 2 grams. If they have low omega-6s, I'll usually do a loading dose that's more than the typical amount, right? I'll do 1,000 or 1,500 milligrams of omega-6 a day for a few months and then cut it back. Let's see. Oh, is there a textbook? Nancy, thank you so much for asking. Yes, there's a new one. And who wrote it? Richard Lord. And it's called Laboratory Guides to Health. Thank you for asking that question. I'm just trying to pull it up here. There it is, Laboratory Guides to Health, Richard S. Lord. And that's the pond or lake right in front of Richard's house. So you can buy this from iBooks or iTunes. You have to view it on an iPad, iPhone, or Macintosh computer. I use an iPad because I don't like Macintosh computers. So it's an interactive and very complicated book. It's 1,200 pages. There's thousands of citations. And the cool thing about the book is that it's interactive, right? So like he'll have a citation and you click on it and it takes you to the study. How cool is that? See, I clicked on that and then poof, it went to there. There's the study. So it's an interactive book. You can read all day long and research, and it's a wonderful educational tool. Thanks for asking that, Nancy. I think it's like $279, something like that. So for this test, for the testing that we're talking about today, you want to do an ion panel or a Nutravel panel from Genova. So we use both single amino acids and multiple amino acids combined. So it's just depending on what the person has wrong. If there's a whole bunch of amino acids off, I'll give a combination formula and then add specific amino acids to balance things out. If someone's on a strict plant-based diet, then I always put them on some extra amino acids. You know, that's a reasonable thing to do. They should also be on some B12 and maybe some carnitine. Okay, there's plenty of ways you can get omega-3s and 6s without eating meat, so that's not a problem. Just have to educate the person about that. If the patient's adrenals are messed up and they have detox and GI issues, I always start with the adrenals first, absolutely. How long does it take to normalize? 3 months, 6 months minimum, you know, I usually retest. Do you get lifetime access to the lectures for the mentorship? It depends on how you buy it, but yeah, you get a long-term exposure to it. You can talk to my salesperson about that, Paula. I don't have any reduced prices for anybody. The pricing on this class is, I know it seems a little bit expensive to you guys, but it's expensive to run this program, you know, and I've worked for maybe 15 years teaching these classes without taking a salary. I only started taking a salary last year. So there's no way we could cut the prices. I wouldn't be able to offer anything if we cut the prices down. I wouldn't have any staff. Let's see, I'm trying to get to all the questions. Yeah, so for omega-6, I use GLA, exactly. How How would you dose low omega-3s? Again, I would start with 4 grams a day for a few months, then cut it in half, the omega-3s. Same with the omega-6s. I would start with 1,500 milligrams a day and then cut it in half after a few months. Before you do the ion panels, you should have people stop supplements for 3 days prior, maybe longer if you want. And then I always do these programs for a total of 6 months. Okay. Is a mentorship program only for doctors? So no, we have doctors, chiropractors, acupuncturists, naturopaths. Sometimes a therapist or a physical therapist will take it. Once in a while, a nutritionist. If you're a nutritionist or a health coach, you definitely need to talk to me first before you take it, because sometimes it's a good fit, sometimes not, you know. And then, okay, one last question. What supplements do you give to someone on a plant-based diet? So I'm going to answer that with the lab. I'm telling you guys, if you learn how to interpret these panels, nobody knows how to interpret these tests. Nobody knows how to interpret these. If you guys take my mentorship and you learn how to interpret these advanced tests, you can be so far ahead. Very few functional medicine doctors out there are trained in these labs. And believe me, I know a lot of people that are just, you know, the majority of doctors that are saying they're doing functional medicine couldn't interpret one of these labs to save their life, okay? They just couldn't. It's rare to find someone. So anyways, oh, plant-based. Okay, so it's common for plant-based people. Some of my best friends are plant-based. My partner, my life partner is plant-based, okay? And I'm not against being plant-based at all. And she's a beautiful human being and she's really quite healthy. But if you're plant-based, you do the tests and you see, and the common patterns you'll see, not with everybody, but with some people, is that they're going to be low in amino acids. Not everybody. Some plant-based people are fine. If they're low in amino acids and you give whatever amino acids they're low in, if they're eating a healthy plant-based diet, none of these things are going to be going on. But I'm just pointing out the obvious. Some people, well, meat eaters and plant-based people can be low in minerals, less likely if you're plant-based, right? But it's possible. And then where you really want to be on the lookout for, because it's hard to get from, you can get 3s and 6s if you're eating chia seeds, pumpkin seeds, flax seeds, almonds. You know, there's plenty of ways you can get 3s and 6s. That's not a problem at all. And then where you're going to see a big problem, though, potentially, not in all vegans, but in some, is with carnitine. You know, carnitine, carne, like meat, right? So some people are fine without meat and their carnitine levels are fine, but some people, like this marker here, will go high if you're low in carnitine. So that's something to look out for, but test, because not everyone's going to have it. And then the B vitamins, especially, as you all know, I'm sure, B12. And B12 is measured right here. So if they're not going to do the lab test, I don't know what I would do for a vegan. I would probably just say take some amino acids, you know, and some B12 and eat a lot of seeds. But a vegan is, you know, even more important if you're vegan to do these tests, just so you're sure that you're getting all the nutrients that you need, you know? All right, let's see, I think we're right on the hour. I'm going to wrap it up. We have this in a recording if you want to listen to it later. And then if you're interested in classes, the boot camp's coming up for telehealth, February 8th, mentorship starting at the end of this month. Tell your friends, sign up, you get $1,000 off if you use that code. And I will see you at the next one of these in the series. I'm sure be getting emails about the next session that we'll be doing. And I'll talk to you then. All right, bye for now.